Okay, welcome back, everybody. It's a pleasure to introduce our chair for the meeting. Oh, if anybody needs Earl Grey tea bags. No. So I first met Kristin, and we were just discussing there. It was in Hawaii in 2000, and it was at an IMA maths meeting. And she was the first person that I ever saw actually present clinical data in her presentation. And so that instantly set her apart from everybody else in the room. Also, her style of presentation made me want to come up and speak to her. And she understands what I mean by that, and you will too. Anyway, ever since then, Kristin's been pioneering, I think, and pushing the field forward. And what's really cool is that she loves her community as much as she loves her science. And that's why it made a lot of sense that Kristin would be the one that would chair this session. So, Kristin, thank you.
That was a much safer and better story than I thought was going to go where we were going. So, phew. All right, we'll make it through this session. All right. So this is a didactic section to tell us a lot about sex differences. And there's, as you know, there's a theme throughout the entire conference on two sets of didactics themes, one on sort of the evolutionary therapy strategies and the other in the theme broadly of sex differences. So we've heard from Melissa, we've heard from Josh, and now I'm going to pivot and show you how these sort of influencers in the community of sex differences have really played out into my understanding of math oncology and some questions that have come up along the way. Click. Nothing's happening. Still nothing's happening. Okay, I'm going to click this one. Take that picture. Okay, the clicker's not. Okay. But also the keyboard is not. Oh, wait, something happened. I don't know if that was, okay, hang on. Oop, oop, oop. Okay, well, I'm going to use the keyboard. I'm going to do it over here. Okay. So sex differences, we've heard quite a bit about this already, and there's plenty to learn about. So I'm just going to blitz over a few things that exist out in the world, right? So we have real sex differences in our understanding of drug metabolism. It plays, you know, you'll run into newspaper articles that look like this all the time. Turns out our dosing strategies we're using for the typical male, and yet when you apply those same thing to women, you can't figure out why these patients don't wake up from surgeries or pick the battle. A much simpler, more accessible challenge here is looking at the average car test. The car test dummies used up until like 2011 was for the average man. And so women were way more likely to be injured in car accidents because of this sort of biased testing. So sex difference is really everywhere in all of biology. And I think you're already convinced of that already. But now you have to, you know, coming from my perspective as a mathematician who was doing oncology research and routinely had a slide at the first of my talk was like, oh, here's two example patients. They're similar. They're this, they're that. They share all these common characteristics. One males, one female. It doesn't really matter though. And I said that line many times in my life, and I was wrong. So really, sex differences is pervasive not only in oncology, but really in all of biology. And biological analyses are going to be are limited by this particular challenge. And let me explain what I mean. So let's just go through a list of things that we have identified sex differences. Gene regulatory network sex differences in normal tissues, looking at COPD with metabolic networks, fine. Looking at colon cancer. We're also picking on metabolism at the moment, metabolic networks. Great. Hence the reason we have things like the NIH policy on sex as a biological variable. The problem is that if you think from the informatics side, if you're trying to make a decision about pathways, all of that pathway knowledge is in some database somewhere, and how things are connected is curated in that way through these grand databases that really underlie the vast majority of omics research. Where you rely on these pathway knowledge. Let's just pick on pathways for the moment, or metabolic connectivity, et cetera. And so these historical databases that really drive, again, everything in the omics world are often male biased, but certainly sex skewed. So for example, here's another database early on in COVID-19 as data was coming together. The lack of reliability of the patient database required retraction of multiple papers because they were pointing in the wrong direction, it turned out, because the database wasn't particularly good and curated to represent all the key variables that needed to be curated. So we put together, with this question in mind, I'll tell you a little story that led to this question in mind. I was taking Josh Rubin back from a sex differences think tank we were running, and asked him this exact question in the car to the airport. I said, Josh, if we're doing pathway analysis, what if there is some baseline knowledge difference between males and females that are not shared, and the database is skewed? And he turned to me in silence for once, turns to me in silence and is like, I don't know what we do about the fact that this is underlying. So I don't know that there's a solution, but this is the community that can answer these questions and help create solutions even when we have biased data in these ways. So the mathematical oncology community is the anti-dogma community, right? We don't have the dogma of, we're not burdened by the dogma that all of cancer is known in exactly this way. We're willing to look at models and play out those models and figure out what works with the data and what doesn't. And so I think that's why this is a natural spot for pitching the fact that we need to be incorporating more of sex differences. And that's why sex differences was a theme in this conference. So I'm going to show you this video that's going to come up next. That's going to explain this in a very fun way. But in short, the underlying concept here is that molecular omics resources, all of those databases I was telling you about, and most bioinformatics tools that then point to any of those databases, really has a real problem, and because of the lack of any sort of sex annotation within them. So if your database is some prism through which you're viewing your data, you're sending in male data and female data distinctly, and let alone TSI along the spectrum of these poles, you put this data through, and your ideal is that you want to be able to, that prism's going to just focus your data and the rainbow's going to ensue and everything's going to be good. But in general, depending on the biases in your data, in the biases in the underlying data within these informatics tools, you can get really wrong answers and fail to detect what is actually biologically there. So this was created by my daughter, and hopefully it plays. Here we go. And okay, that's the end of my talk. So that's COVID era work right there. So my daughter was asking me what I was doing, and she was playing with iMovie, and there a miracle occurs, and this is how you get it. Okay, but the point there is that underlying a lot of what we're trying to do is not only the challenge of saying that there's sex differences and that we want to go see them, but two, the tools that we have available to us. We really have to be critical about assessing what's inside those black boxes, whether it's an informatics platform or whatever we're building, and whatever those core assumptions are, which are true for any mathematical modeling that is done, including on the informatics side. So I'm going to use this background to now pivot to the discoveries we've made in the context of brain cancer. We and others have made in the context of brain cancer, and how that thus all connects together in the theme of sex differences. And I have to say, I say this almost every time I give a talk in this spirit, that once I started looking, I couldn't stop looking, because there are sex differences everywhere in at least our data. So I encourage you to look. So the brain cancer that obviously we focus on is in glioblastoma. It's a deadly cancer and neurological disease, because it's so deeply integrated within the normal brain of single cell invasion into the brain. It's a fairly rare-ish cancer. It's not as common as things like breast cancer, but it's so deadly. With very aggressive, even with very aggressive treatment, 5-year survival is, you're very lucky to say 5%, and we're not moving the needle. We're not making progress, sadly, and we need to do better. And so everything in my lab has always been focused on this concept that really every patient deserves their own equation. So this idea that every patient should be individualized. And I think we all believe that, but we also need tools to be able to deliver on that idea. So here's 15 newly diagnosed glioblastoma patients, all receiving the same exact therapy. And look at that diversity of response. Those 15 patients look the same under the microscope. They might have different sex. They look the same under the microscope. They have all the key characteristics that you would attribute even to this day to them as the same, and yet that's the heterogeneity of the standard of care. And that tells us everything I think we need to know about how poor we're doing for these patients. These couple of patients that respond a lot, okay, great, we did something for them, but these patients at the top we're not, and we need to be able to know who those patients are. So, sort of foundational work within our lab has been to break down the biology into these simple mathematical models, and use those mathematical models to inform how to pivot and reveal insights into the kinetics of each of these individual patients' disease that we can leverage for treatment. So here's a patient's brain segmented out of one of the patient's that patient abnormality, the extent of what you saw on MRI that Pam was telling you about a few minutes ago. And if you zoom in at a single voxel level, this is what you see. It's those individual red tumor cells. Well, they're not red in real life, but like they're red in the video, tumor cells that are invading into the black background. And that black background is normal brain. There's neurons there, there's oligodendrocytes there, there's astrocytes there. They're probably reacting to that invasion. But that gradient of cells is not a ball and clumps. It's individual cells doing their thing. Now, so as a mathematician, of course, I put my mathematician's hat on this, and I can track those cells. I can write down some equations about what describes that behavior at a phenomenologic sort of high level. So I've got dispersal of those individual cells, I've got proliferation of those cells, I've got response to whatever radiation, whatever treatment they're receiving, and then sort of the rate of change. So you can build these really simplistic models, and you can get really far with that. And you've probably, if our work in this area has been decades focused on the idea that you could then use patient MRIs to personalize those mathematical model parameters for those individual patients. And then you can go from, oh, well, if the MRI is here, the red abnormality, can I tell me how many cells are there? Because I know there's some cells there, but how many are there? So you can start filling in those blanks and then sort of personalizing these sort of patient-specific predictions of even treated tumor overlaying treatment into those models. Fantastic. This is a great baseline upon which to sort of build an individualized narrative of what is going on in each individual patient, as opposed to after the fact plotting the graph and going, well, I think we did okay for those guys, but not those guys, right? So over the last 20 years or so, I've built a very large database of these patients. We've got over 5,000 brain tumor patients that have received a diversity of at least 140 different treatments. There's 100,000 MRIs in this database. Over 50,000 of those MRIs have already been segmented. And when I say segmented, it means I can recreate that three-dimensional pretty picture with the red abnormality. And so I can tell you exactly where in the brain there is. So that becomes an interesting data problem. Now it's a data opportunity to now go look at things through a different lens. So we can track the serial evolution of that imaging abnormality, but we can also just start asking key questions kind of at almost a population level about those. And what key question are we going to lead with? Sex differences. So First of all, we knew this in, I'm not showing our cohort here, but we know this in general, right? SEER data shows this, as does many multiple, smaller cohorts within institutions show that there is not only a sex bias to the incidence of the disease of male skewed 1.6 to 1, but the survival is beneficial to females. Okay, so there's obviously a sex difference at the outcomes level. Are there sex differences in the biological level? This is work with Josh Rubin, where his team was focused on looking at transcriptomics and doing I was going to say the biases associated with all the databases, that I was referring to before, was using a method to be able to sort of separate out the signal, because there's going to be a lot of shared signal between the sexes. There should be, and so this method known as JIVE, is Joint and Individual Variance Explained, is an opportunity to take the shared information, subtract that out, and then you have two sex-specific remainders. Now, you can look at the sex-specific remainders. There's overlap, obviously, in these situations, but there's also distinct components of the transcriptome that comes out. But if you look at the clusters of female-specific, there's like 5 clusters. You look at them and you go, okay, well, let's pick out the interesting cluster. The interesting cluster is presumably the patients that live long. What explains the biology? What a biology is associated with those patients living long? It's integrin signaling. If you do the same thing on the male specific clusters, look at the 5 clusters, pick out the cohort of patients that live longest, and you go, okay, what's the long-lived male patients? It's cell cycle control. So there's different biological mechanisms. This implication of this is there's different biological mechanisms that connect biology to outcomes, to improved outcomes in this disease. Now, taking that concept and now going back to our patient imaging, this was the glue that we were trying to provide. We have the same model that you saw with the red movie, and we're estimating those model parameters in green, and we're going to connect those to overall survival from patients in our large cohort that I was telling you about. Extreme survival is a small subset of patients that we have that live over five years. We have a large study of a couple hundred of those patients, or patients that are short survivors, live less than 6 months. And no matter which band you choose, the mapping between those survival elements and anything that we can infer from imaging is different between the sexes. So the imaging patterns that predict outcome are different between the sexes. Like, okay, fascinating. Again, you keep looking and can't stop unlooking. So next up, here's just another kind of visual of what I just showed you on the previous slide. This is taking a subset of our large cohort of patients, and just looking at the imaging pattern of diffuse invasion. So invasion matters for women, but it doesn't matter for men. The pattern of invasion that you see on MRI matters differently. And these are the ones, these are the estimated low invasion versus high invasion is estimated from our mathematical model that you saw in those previous slides. Now, everything I've talked to you about here in terms of, the biology of the tumor has been pretty simplistic. It's one sample per patient gives you a transcriptome, or one measure of how invasive does that look on that overall MRI. Okay, fine. But the biology is, of course, much more complex. There's a ton of intratumoral heterogeneity in this disease. So the way I like to analogize this is when I think about the intratumoral heterogeneity, I think about the MRI being a map of the human brain, right? So it's like a satellite map, how to view onto something that I can't touch. So it's just a satellite map. So now let's do a satellite map of a country, and let's go in and we take a piece of tissue and we get an answer, and the answer is red. And now we've got a drug for red, and we're going to match those two things up, and we're going to have precision medicine. But that's the problem is the biology is, of course, to this community completely clear. It's much more complex. There's reds, blues, probably purples and rainbows, and it evolves over time. So really the future of precision medicine has to incorporate all that heterogeneity. And in the case of glioblastoma, really understand what are we leaving behind because of that diffuse invasion. So I'm sampling whatever I'm sampling surgically, but there's always cells pioneering off. And how do I even get a guesstimate at what those cells are doing? So that's why we've really leveraged a lot of focus on these image localized biopsy strategy within our lab. So you've seen a couple of presentations that have discussed this, including Lee's this morning, where it's very much detailed the protocol. This is another patient example. He had a cooler video. I want his video. Jaslyn, thank you. This is another one of these patients that came in. This patient was seen at an outside community oncology community oncologist and was referred to Mayo Clinic. When they came to Mayo Clinic, it was 17 days later, because I think they came from out of state, if I recall correctly. And by the time they came to Mayo, you're going to see the imaging abnormality grow significantly. And then the patient was sent to the OR, and immediately we collected, I think, 11 biopsies on this particular patient at lots of different locations. So this protocol that you've now seen, we have now over 1,000 biopsies for over 200 patients. I mean, there's a ton sitting in the freezer because there's never enough resources to actually process those tissue properly. But we also have a broader collaborative, like a consortium that we've created with other institutions that are approaching these biopsies as well, to try and build this out even more robustly. So one of the things that you can do with this sort of data is you can take a single cell annotation of brain tumor specimens and break it down into the copy number aberrant cells, which are presumably the tumor cells, and you can classify them however you want. We're classifying them in this particular visual. It doesn't really matter for the story that I will tell you. And then you can break down the non-tumoral cells, the neurons, myeloid cells, endothelial cells, reactive astrocytes, oligodendrocytes, oligodendroglial precursor cells, all those cells. And once you have those individual single cell signatures, you can use tools like cellular sort X. There's a ton of deconvolution tools, but again, it's an AX equals B sort of problem. So you can just infer what the relative abundance of those individual populations need to be in order to make the bulk signal make sense. So then that's the heterogeneity that you see across each of those samples. So you'll notice that it's widely heterogeneous, number one, but how can we use this information? Well, one of the first things we can do is we can separate the males and females and simply ask, when one population of astrocytes is around, is that kind of population, that the typical type of glioma cells that are typically adjacent to those type of astrocytes also around? It turns out those patterns of interaction between those cellular populations seems to be different between the sexes. And this is new work where this is very early, but this is what we're hoping for funding for in this general direction, where we're focusing on looking at deconvolving the entire cellular environment and understanding what are the cellular interactions that are then creating the imageable niches that are manifesting on MRI that I just showed you in a couple previous slides are not the same between the sexes. So we know that. So this would help explain that connection difference. So let's pick an example of a patient. Here's a patient with 7 biopsies. I've just color coded to you those same cell populations. I just showed you on the previous slide, the relative abundance. And you can see that you sample 7 areas, some really close to each other, some very far away from each other, and they're just very different. There's a lot of heterogeneity of what cells are where and what they're doing in collaboration with each other. So we use data like that to not only think about how to build a tumor, how to build complex models of the complex cancer system at the cellular population level, but also how to glue that information to the MRI. So we take the image localized biopsies. We've got this, whatever analysis we've done. I'm showing you transcriptomics. We've got exome. We have a variety of projects to do a variety of things. We have immunohistochemistry. And we line it up to the image localization. And then we read out all the local regional imaging information, whatever it is, some sort of grayscale understanding of what's going on in the image itself, in the region where we said this piece of tissue came from, and we align it to whatever biological information we have, which could be cellular populations, it could be anything. But in our hands, a lot of it right now is genomics. And we fuse that together to build these mathematical models for predicting some aspect of biology. You heard her talk about this from Andrea that was looking at T cells, and you're going to see a little bit of that in a little while. So basically, we want to fuse a model that then now allows us to just take the entire satellite image of the brain, the MRI, and tell us what the biology is everywhere with some certainty, right? What that biology might look like, and how to then understand how that entire landscape is changing during the course of treatment is the focus of this conceptual work. Now, the problem is, again, I showed you other data. Let's look at the cell populations on the bottom. Those are just those deconvolved cell populations I've told you about. And those are the imaging, just standard signals, like the mean signal, how bright is it? And the variance in that region, right, very near it. And you see that objectively on the left and right, the mapping between what you see on MRI and the biology on the bottom in terms of cellular abundance of those cell populations is different. And that cellular abundance is inferred by this deconvolution approach that we were just telling you about. So again, at every level you look at, there's a difference in the mapping that we see that is sex distinct. So the problem is now you want to take an image, patient image, and you want to predict a map of what the biology is over the whole image. So you do so, and you train your models, and your models are really hungry because these are machine learning models. They're desperate for data. So you want to throw in all the data and maybe add a category, add a covariative sex, maybe, but it's not going to work. It turns out it doesn't work at all. The only way you can get increased accuracy out of your models, it seems, is that you separate the data completely. Because those patterns are just distinct enough that there's not enough, that the shared information isn't gaining you, at least at these numbers of samples we're using. So if you look on the bottom here is basically the model accuracy as a function of whether I used fewer samples, male pile, female pile versus combined, most data combined. Intellectually, machine learning should be happiest over here. It's not. It's happier when you've separated out the two piles because the signal is just that different. The accuracy tends to go up even though you're giving it less information. So we focus on all of our approaches now that are trying to fuse in the MRI data down to the biology. We fuse in this knowledge of sex as a biological variable. So how do you use something like this? I'll give an example. Here's 3 patient examples. So these are 3 patients that were on a clinical trial that was focused on targeting EGFR. This drug actually targeted erlotinib. I think all of these patients were treated with erlotinib. So these 3 different patients, one progressed, one had a kind of stable disease from that start of treatment to 3 months in, and one had response. And using our sex distinct models, we can overlay prior to treatment whether the target, in this case EGFR aberration, was present, and it was highly present in what turned out to be the patient that responded, and it was not present in the one that progressed, and that makes sense. So this is exactly how the rubric about how you would approach using this, but again, the accuracy of your model is strongly dependent on your incorporation of sex properly into the, I'm not claiming that we've done it properly, but sex should be incorporated into that story. So this is just a smidge of overlapping with what Andrea was talking about the other day. This is a slightly different model than what she presented. But if you look at the case of immunotherapy, it's this real challenge because this actually upsets me every time I tell this story. Is you're sitting as an oncologist and your patient comes in with this at -1 month with some image that looks concerning for progression. You do a short interval follow-up at month 0 and you're like, no, that's real progression. Now let's put you on a therapy. In the case of immunotherapy, because of the delayed time standard for which you have an evolution of response that takes a while to show up, and it looks a lot like tumor progression for those next 7 to 9 months, that patient that at month 0 had a median survival of 4 to 6 months has to wait through and hope that what is worse from an imaging perspective is better for them longer term. It's a real confluence of ethical and every challenge under the sun. So how do we do a better job of knowing very early that this patient's going to turn course at month 9 and go back? And so we were really, a ton of my lab resources now is focused specifically at this question, hence the reason we're interested in that cellular deconvolution of the environment, because the answer to this is not whether EGFR is present or not, as I was showing you in the previous slide. The answer to this is what immune populations are showing up and who's leaving. So can we do a better job? Can we avoid this really actually, integrated into clinical practice? The IRANO, immunotherapy for response assessment and neuro-oncology assessment time point is 6 months, expecting you to say, don't make a change in treatment course for this patient for those first 6 months that a patient's on immunotherapy, because we really can't tell you whether they're responding or not. So again, it's a real ethical challenge as to manage patients in that context. So there's a ton of immunotherapies out there. I remember being a graduate student and, we thought there was nothing immune going on in the brain really. But now we're doing all this crazy immunotherapy stuff and we're putting it into patients' brains, including things like dendritic cell vaccine trials with our collaborator, Ian Parney, who's up at Mayo Rochester. So we don't know enough about what the dynamics of the immune system is within the patient's brain, and we're trying to apply these technologies that are perturbing it. And so we need to figure out both baseline what is it doing and what the perturbation is doing. So we looked at the pre-vaccine and post-vaccine imaging. I'm showing you a separate model than the one that Andrea showed, but we're basically overlaying. If a dendritic cell vaccine is working, then basically immune cells are going to T cells are going to show up and they're going to do something. So one potential indicator would be T cell abundance. So we basically built a T cell model using the pipeline that I've showed you over the last several slides, and we've sort of overlaid that on these patients, and we looked at their first imaging follow-up time point. So only 2 months in, so patient receives the vaccine, and then 2 months in, they get an image. Now, the patient on the top is interesting because the imaging abnormality shrinks. There's less of it, right? And so from an imaging perspective, you're like, oh, this is good. This is a response, but it's too early for an immunotherapy response generally. But that patient ultimately only lives about 5 months. Whereas the patient on the bottom doesn't really respond, stable at best, kind of sitting there, looks like smoldering, and they live 37 months. But the difference is we're overlaying, so the extent of the blue that I'm showing you there is the extent of the imaging abnormality. If you overlay in the color is the sex-specific radiomics model for predicting T cell abundance. And you see that the T cell abundance seems to be going down for the patient on the top that only lives 5 months, and the patient on the bottom is 37 months. So now you've got 20 of these patients, you're running a clinical trial, what do? On average, the trial was a failure, but could we find early imaging biomarkers that kind of separated out the failure, the overall average failure, to this individualized heterogeneity across patients? And basically, this was the first analysis that we did. We found that basically, if the pattern was the way it was going up, there being more T cell signal was present, then the patients did well. They ultimately actually responded very well and had very robust long-term outcomes that were measurable in not only many months, but could be used to decipher how to revise the next phase of said trial and provide evidence for why to move forward with the trial. So taking all of this together, I hope I've compelled to you the reason why, one, understanding sex biases in not only the platforms in which you rely on from like an informatics tool set, all the way down to an image. I mean, why would you think the MRI would be different? Like, I'm still blown away that like, why is the biology underlying that distinct enough that I'm seeing this loud of a signal this early? I think that those profound challenges are happening every day in the clinic, and it's our duty to stand up and contribute to solving that very straightforward problem. So I want to thank everybody for the support that's been integral to all of our work, and thank everybody, many of whom, this is a very old picture, unfortunately, because I uploaded the wrong slide. But this is a subset of the team. There's several folks from the lab that you've been hopefully circulating around within the group. Thank you.
Fantastic talk. I'm thinking back about Josh's presentation yesterday. There is no dimorphism. It's more of a kind of continuum. Yep. I assume there must be an overlap where the patients would make categorize closer to male, female, yep, to male, and the other way around. There may be some distinct features of reasons. Are there any kind of imaging-based biomarkers where one can place the patient to the kind of more appropriate being?
Yeah, could we basically take the TSI and convert it to an imaging and look at the mapping? Yeah, no, for sure. I think that's a great project, and we were talking about it last night and today already. So yeah, no, I think it's an absolutely spot on project of where the innovation in this area is. I would have to say I have a funny little side story, and I don't know how much this was a confluence of a summer project. We had a summer student come in, like I think you were from Stanford or something, like for just like an REU something, and they were assigned the project of trying to just build a simple machine learning model. Can you separate out sex? And it was mind blowing because it was like, with very little data, they were like, oh yeah, well, that's a male slide, that's a female, and you're like, whoa, this was the early days of this. So I think you're right. I think there will be something there, but TBD. Talk to me in a year.
Hi, thank you. Thank you so much for the talk. That was so interesting, and I found the the slides on reconstructing the interaction networks particularly interesting. I was wondering, when you've got these kind of incredibly heterogeneous populations, even within each sex, generally the statistical problem of reconstructing an interaction network is kind of challenging, and there's this statistical phenomenon, right, where if you have a heterogeneous population, you could just subset it randomly and then reconstruct two completely different interaction networks from a population that's been split randomly. So I guess basically what I was wondering is, do you then within each sex, for example, split your female population in half, reconstruct two interaction networks from each half the data, and see how well they agree?
Yeah, I did not focus on all the statistical tools that one uses to be able to do so. I think the key message here is that if we weren't looking, that would have just gone under the bed. And I think that's the call to action, right? From a didactic standpoint, is that I was definitely not looking, for years and years and years, and then I saw that guy talk, and then it made me look, and now I can't unlook. So I think you're absolutely right that there are components within this, and yes, we take into account those in those creation of those interactions for sure.
No, so I haven't seen these differences before, so this is mind blowing for me as well.
Thank
you so much.
Great talk. For a long time, I had a question about whether the degree of microenvironmental heterogeneity is informative for response to therapy, time to progression, et cetera. You've got these nice microenvironments in the cyber sort. Do you see that in GBMs, some quantification of heterogeneity?
Absolutely, yes. So we actually have a lot of metrics of sort of those. We have several metrics that do look at diversity of the environment and how that contributes to things like response. The first is the diversity amongst the samples within the patient, right? And you've got to control that and then go beyond that. I think there's a lot of data to do a lot with, so we should definitely talk about more stuff. But we have little. I just don't have it on like immediately at hand. Yeah.
Very inspiring stuff. My question was a little bit on the early part of what you had mentioned. So with the predictive model, right? And once you find out specific expression level differences from the transcriptomics, how do you put it back into the model? And you said like, you can change the diffusivity term or the growth rate, but what are those relations, and how do you navigate that? Because that's
it's absolutely, it's the holy grail of what my lab wants to do. So so this intermediate point of the heterogeneity story, you're not going to we don't know the answer we don't even have really good models, experimental models, to tell us what that answer should look like. So using this image localized biopsy data to deconvolve the environment, we can then look at the interactions between the populations and then use that to build a more advanced, more refined version of that model that I was showing you at the very beginning, which was basically proliferating cells that also invade. That's it. Right. But there's a lot more to it, right? So it turns out the reactive astrocyte population is huge, and there's like a whole story there. We have a UL1 that's focused exclusively on the monocytic sort of origin cells, so macrophage monocytes and microglia, and looking at their relative phenotypic state and how that drives the tumor in different directions, particularly under immunotherapy. I think we partner with like six immunotherapy clinical trials to deploy models just like this, but for those subpopulations. We have this work with the T cells, and then the holy grail is you get enough models, because not all models are going to work. Like just because I want to see that cell population doesn't mean the MRI is going to tell me, right? But there are going to be some of them that are going to work, or some combination of them that are going to build successful models. And then you can apply it to that huge database of patients where I've got serial imaging, and now I can go run forward in time the biology of whatever piece that I can say the MRI is reading out with some authority in a sex specific way. And then they'll use that to build spatial temporal models and not only inform those spatial temporal models in terms of design, but also validate them. So that's the closed loop that we're working towards.
So cool. Yeah.
Pray. That's the holy grail.
Thank you. That was truly inspiring. So one of the things that I'm always wondering about is like, how can we combine spatial information and temporal information, especially because the fantastic methods that we have for spatial transcriptomics are destructive, so we cannot do this here. So it looks like to me, or maybe I'm just seeing the hurdle here, that because you do have resections of tumors as well, right? Could you do spatial transcriptomics on that and then correlate that with the biopsy and MRI data and really see like, wow, what's going on in space and time? That would give me chills.
So okay, come along then. So we had in our grant application that's under review right now, we're doing exactly that. So we have spatial transcriptomics on specimens for which it's not the example from Lee. You saw the picture where it was like he had the little tiny little ditzel, and then you had like the little bigger piece of tissue. On the pieces of tissue like that, we work closely with the chief of neuropathology at Columbia University, a guy named Peter Canal, and he has on those larger specimens spatial transcriptomics, and you've got the same cell, the signatures for those single cells, and you just simply align them and overlay them and understand what is the degree to which this tiny scale is manifesting to the MRI biopsy scale, which is, cubic millimeter-ish, scale, and what gets you up across those scales. So it's the other way of bridging all of those scales, because that'll inform you that then you can put that in your mathematical model and see if it goes forward, and then check on the image, because you've got some trained model that tells you, no, that's the T cells aren't there, or whatever the biology is that you're looking at. So yes, I'm super excited about doing exactly that, because I think that's the only way you're going to close the loop on something so challenging as brain tumors, because you have such limited access to tissue, period, end of story. So you really have to reap as much benefit as you can from what little tissue you do get.
Wow. I mean, that sounds like sci-fi almost.
Well,
Do you think in the spatial transcriptomics data, you could potentially pick up resistance and then see if, that would be
I think for sure you could see signatures of what is a conducive environment for response versus non-conducive environment for response. Resistance to me is like a trigger word in the context of glioblastoma for me, because they don't respond all that much on average. And so for me, I'm like, maybe we're not getting them, in the first place, because we don't know who they are.
Okay. But even if it's like on a continuous response scale, I think that would be super, especially in context of depth of therapy.
Yeah. So specifically, that grant has perturbation studies. So we take the spatial transcriptomics, and then we have adjacent slices, and then we do perturbation studies on this brain slice assays, and perturb the environment, and then redo the spatial transcriptomics, do spatial transcriptomics on that adjacent piece. Yeah.
Blows my mind. Thanks.
I'm wondering, do you think the sex-specific differences might be more or less pronounced in the brain than other tissues?
Oh, that's a good question. That's a really good question. I don't know. I asked the question of a poor summer student, thinking that it was going to be just like one thing to get an answer. He would get an answer in the 10 weeks that he was there, instead of just blowing my mind. And now everywhere it seems to be a signal. So I don't know, but it's a great question, but we should ask. All right. Thank you.